This is a production of Cornell University.
Thank you, Margaret, for the introduction, and thanks for the invitation for this talk. So yes, I'm calling in from IST Austria. In more exciting times, I could have maybe visited Cornell, but now it's you have to do with the background of my fake background, actually, and then what you see on the slide. So IST Austria is a multidisciplinary institute. It's doing pretty good on the world ranking, but that's not what I want to brag about. It's multidisciplinary, what makes it very interesting. There's actually only two plant groups at the moment, which is my group and then Eva Benkova, who works on cytokinin group. So it's Hirsi's group and Eva's group. And in 2022, there will be an extra plant group, but that will be on epigenetics. So the institute is most known for the interactions between other disciplines. So then what I would like to talk to you about today is the rapid responses in root growth. And that's actually exciting research. What has been playing the whole time in my background here is this rapidly growing root. Well, it's actually just a normally growing root, but it is actually a demonstration of what we have changed, how we can image roots live, and what technology we have done to do that, I will introduce a bit later on. So we can actually live see how a root grows and whether it interacts with other roots, like in this case, or whatever other thing that it would come across. So I divided this presentation in two main subjects. As Margaret said, I have actually two papers under revision. I made this kind of timeline that you will see in the top of all my slides, so you can follow where you are. And if you would have questions, please then type them in the chat box. I will try to handle them at the end or in the student meeting afterwards. So in the divisions, you can see I will give a short introduction on Auxin. Then, as I said, we have exciting technology, and then it will move on into the things that we identified. So there will be AHAs, which I will tell you what these are, and then there will be a part on myosin. And then everything highlighted will always show you where we are, so I hope it's easy to follow. So Auxin, I don't know whether it needs a big introduction, but it was actually the first discovered plant hormone, and that is because it has a very strong growth effect. Like almost all developmental plant steps are actually affected by auxin, but the phototropic and the gravitropic growth are mainly regulated by auxin. And because of this phototropism, which was a clear thing, already in the 1800s, people discovered that there was actually one molecule or a family of molecules responsible for this that was transported in the plant that could be leaking out of the coleoptile tip, which then, if you would put the agar block in which the hormone had leaked again on the coleoptile, it would still do the same thing. So the coleoptile that would sense light would grow towards the light, and that's still the case if you replace it by a block of agar that just contains something. What is already a long time established is that this bending is because of this molecule auxin, and it's actually auxin that gets redirected. So you have it in the beginning symmetrically distributed everywhere throughout your system, but depending on an external influence, you would have it asymmetrically being redistributed, and then you had to have cell elongation happening. So this is what I said. 1830, auxin got his name, and auxin comes from the Greek auxin, so it's actually to grow, and that's where it got its name from. Most of the time, it's made in a plant based from the amino acid tryptophan, but there's actually quite some pathways known that don't involve the amino acid tryptophan as well. It's just the major pathway is tryptophan dependent. It's not unique to plants. You can also find it in green algae, or at least some of the biosynthetic enzymes are also present in green algae. So that's the lowest class where you can find it, but even some bacteria, and mostly soil bacteria, have auxins as well. Endogenously in a plant, you would find this molecule most, and that is called indole triacetic acid, sorry. And that one is found most, but there's actually these other three that, depending on the species, you can determine as well, or it has an auxin effect as well. Besides from this endogenic auxin, you have a lot of synthetic auxins, and I'm introducing them because in this presentation, I will both use NAA, which is one of the synthetic ones, and IA. So that's why I'm introducing this. What auxin is most uniquely known for is not just that it affects most of the growth responses in a plant, but that it is transported uniquely. It's, I think, the only hormone that is actually between cells in a unidirectionally way transported. So what is happening if you look at on the plant scale, you have actually auxin being produced in the shoot apical meristems and leaves, also in the buds. From there, it's loaded into the general transport pathway, so it goes into the phloem down towards the roots where it mostly would act, or down towards buds that it's inhibiting from outgrowth, so it's transported downwards, general transport pathway. And then when it comes at the tip of the root, there's this fountain pathway back. Now, all of this depends on actually intracellular specific transport of auxin. Otherwise, you would never be able to go back up from the top. And that is depicted here. So what happens with this molecule? In the apoplast, which is slightly acidic, you have the molecule charged, and that actually can enter into a plant cell through diffusion, so just passively going in, or through influx carriers. They are known, we know what these are. Then something is happening inside of the cell. No one exactly knows what yet, but what is happening to the molecule is that it definitely becomes charged, so it dissociates from a proton, and it will need to get out of the cell, it will need active transport. And for that, there's actually a big family of efflux carriers known. They are called pin transporters. And this is where my boss here became famous with. So he is the discovery of the pins, or the lord of the pins. And these pin carriers is actually what makes auxin transport uniquely unidirectionally, because they are not just randomly placed within a cell, they very often go to a specific location, as you can see here. So these images are actually from immunolocalization, which means that you're not looking at the GFP, but at the endogenous protein. So you use an antibody against the protein that then you can visualize using a secondary protein. So this is the normal localization within a root of pin 1, and it's basically in all the cells. And then if you would look at pin 2, another pin that is actually together with pin 1 responsible for this fountain transport, you see that because of its localization in other cell layers, apically, you can actually establish this fountain. So as said, this is unique. What is then also important to know is that auxin signaling is known forever as well. And since the 80s, people started to temporarily dissect this process, and the closest or the fastest responses that people have within the signaling pathway is within 2 to 3 minutes, you have actually sour genes going up. Growth, however, is delayed, and it happens approximately 10 to 20 minutes after you apply the auxin. So there's actually a delay in those things. So primarily it was thought that auxin regulates transcription. And that happens through this. It's the simplest cartoon that I could find. It's actually a funny one, because what you have is you have receptors within a plant. The signal, of course, would be auxin. And when auxin binds to the receptor, it actually attracts within its complex a co-receptor, and that one thereby gets ubiquitinated. It gets targeted for degradation in the proteasome, and it relieves this activator. So suddenly they can be active, and then transcription of proteins can happen. So this is how it looks a bit more complicated. And more interestingly, this is happening in the nucleus of the cell. So you have these TIR receptors, which are waiting for the signal. Whenever auxin is coming, which is this green molecule, it attracts these small repressors, which are called AUX-IAs. Within the complex, they get ubiquitinated, they get degraded, and the ARFs are able to initiate transcription. So this is what's happening within the nucleus. However, things are already known for a while that are faster, and they are also auxin mediated. For example, there is the recycling of these, and I just introduced them to you, these PIN exporters from the membrane. They get recycled and back to the membrane, and that is also a process that is affected by auxin itself. So that is a faster process that most likely does not involve transcription. Then there's also another possible pathway that involves a transmembrane kinase, and that one also might regulate transcription differently. And then lastly, and recent work has shown that what was thought to be a purely nuclear signaling pathway with these TER-EFPs residing in the nucleus is probably not entirely true, because within the family, you actually have members like this AFP1 that are cytoplasmic one. So you probably also have receptors within the cytoplasm. So if you would look at a signaling pathway in a logic way, what would be is, and especially as I told you, auxin is unidirectionally transported within cells and comes from the apoplast, so from outside of the cell. If you would look then at a signaling pathway, you would actually have a hormone from outside binding to a receptor that potentially should be residing within the plasma membrane and then generates a signal transduction within the cell. So this is what you would expect. What I just explained that we know from auxin signaling is nothing like this, because we don't know how this part happens. We don't know what happens within the apoplast, within the cell membrane. We only know what happens with the receptors in the nucleus itself. So there's actually a long-going discussion and even questions being asked for a long time already, whether there would also be an apoplastic auxin signaling pathway that then mediates fast signaling. And there I have to touch a controversial subject within the auxin field, because in the beginning of 1980s, a protein was found, and it's called auxin binding protein. And that protein was found to bind with high affinity to auxin. And it was found to be residing in the endoplasmatic reticulum, but also in the apoplast. It is controversial because at some point the mutants in this protein were not what they were supposed to be. So they also had a mutant in the neighboring gene. So all the phenotypes were not correct. But we still do think that maybe there is an extracellular pathway mediated by ABP1, because that protein is there and it has high affinity to auxin, and then probably interacting with leucine-rich repeat receptor-like kinase co-receptor complexes, whereby it's mediating intracellular effects. For example, ROP-RIC signaling is shown to be affected by a pathway like this, and it's important for PIN internalization. So again, these pins come back, so these transport molecules, they get internalized to be recycled back to the plasma membrane. So for that, auxin has an effect on that pathway, and that is a fast pathway. It's not involving the nuclear transcriptional signaling that we know. And then secondly, because here it's still vague, it just says leucine-rich repeat receptor-like kinases. Secondly, and recently, there has been a paper that actually gives a name to one of these potential co-receptors. It's a transmembrane kinase 1, TMK1, and in the closed apical hook, it's a very important protein, because when actually the hook has to open, or it is actually deregulating the opening of the hook, then there's a auxin signaling triggering this TMK. And the kinase is actually a very weird one, because it releases its kinase domain, which goes to the nucleus, there phosphorylates AUXIAs, the repressors that I talked for before, and then make sure that differential growth can happen. So yes, potentially there is an apoplastic signaling pathway, but we don't know really what is going on yet. Then, as I said, we have things happening within 3 minutes transcriptionally, but what we actually do see is that, for example, a calcium spike upon auxin happens within a minute, which is much faster than the 3 minutes. That cannot rely on transcription. There's other things like growth inhibition that happens very fast as well. I will show you that later. So we needed to develop microscope techniques or microscopical techniques to be able to actually visualize what is happening very fast upon auxin treatment. And in our group, we have this unique system that combines a vertical microscope with a microfluidics system. And the vertical microscope, I will show you later, is just a flipped microscope. So it allows you to image plants within their physiological conditions. So the root goes down and the shoot goes up. But it is also combined with an automated tip tracking system. So we don't have to measure growth as such. What is happening is that in a MATLAB-based script, we actually determine what the position shift is between time point 0 and time point 1 and time point 2. And based on these two calculations, we calculate where the window has to shift. And you have already seen a result of that, because that's why in my first slide, I can image this root growing the whole frame, because this is high confocal microscopy imaging without having to change the microscope. So this is all happening automatically. The system calculates how much it should shift, and it shifts as well. Based on the coordinates of the shifts, we can then calculate the growth rate. So it's actually very convenient for us. And this is how it looks like. So we have actually really literally flipped the microscope with 90 degrees. We provided some extra support so that you can actually lean into it when you have the screen, because you have to stand at this microscope. And then you have at the stage, you actually have just physiologically, as I said, the roots growing down, and you can image these over time. So we can use confocal lenses for this. However, any high numerical aperture confocal lens would require an immersion solution. And as we have flipped everything, anything you would apply here would drip off as well. So we are kind of limited to expensive dry objectives like a 40x objective. So we can't really go into like cytoskeleton details in this system, but we can do very good imaging. Then I said we do a combination of things. So we also, to be able to visualize very fast changes in treatments, we also have developed this microfluidic system. And it's actually just a polymer in which we have channels left out. And the channels are super low volume, which means that if you want to change treatments, it goes very fast. And it's actually controlled by an airtight valve. So you have a valve that will close when you put air through it. And then when it opens, you have the medium streaming through. This allows us to image 8 roots at the same time and all individually treat them or don't treat them. So this is how it looks within the channel. The root is nicely there. And this system works very well. But why is this all important? Like every single one person that has worked with auxin knows that auxin is actually inhibiting the root. Like you see, a normal seedling at 4 days looks like this. An auxin-grown seedling has a very tiny root and has more root hairs. This is how it looks like in our root imaging system. So what we do is actually we have this root growing in these microfluidics channels. And every time you see the background turning pink, that's when we have auxin in presence of the root. And you kindly see it because it's actually growing, stops growing, growing, and it stops growing. So if you would plot that now in frames, instead of looking at a movie, you would actually see it's standardly growing, it's generally growing, and then when you apply the auxin, it's inhibited, and when you wash it out again, it's growing again. So this allows us to really image root growth. And when we saw this, we knew that we had the system ready to really visualize very fast responses, because what we now can do, we can actually look at what is responsible for auxin to do this root growth inhibition. And that's the first part of my results then. What we did is we looked at responses within a plant that would correlate with this root growth inhibition. So we looked at, are the microtubuli affected? Well, they were not, or at least not within the same time frame as we saw the root growth stopping. So probably they were not the cause for any of the auxin effects, of the fast auxin effect we saw. We looked at vacuolar structure, that was not affected either. But then we shifted to this. We actually, and that's also because of a long-lasting hypothesis that I will come back to later as well. We actually looked at what is happening with the pH around these cells that are stopping to grow. So for that, within our system of the microfluidics, we just had to add one more dye, and it's a ratiometric pH indicator. So it's a dye that you just can visualize. And with that dye, you can actually see what is happening in the apoplast of cells. So same idea, however, the frames are now within 30 seconds of each other. So what you would see is 30 seconds, 30 seconds, 30 seconds, nothing is happening. At time point 0, we apply the auxin, and then you suddenly see that the color shifts, which means in our case that the pH is increasing. So we actually have almost immediately an alkalinization. So it's getting more basic in the apoplast of the cells. And if you would quantify that, you actually have a super nice correlation graph between growth inhibition, that is in black, and then the pH that goes up within the roots. So we think that we have found alkalinization to be the cause for this rapid root growth inhibition. And then we dug deeper into this, because we had a causative or correlative effect at least. So what we then did was, can we mimic this by, instead of doing auxin, can we already change the pH in the external medium of our roots and then see what is happening? And actually that worked very nicely, because what we do then is we have our normal medium that is pH 5.8. When we exchange that for a more alkaline medium of pH 6.15, the root stops growing and it retakes back its growth when you switch it. On the opposite is also true. When you exchange your medium for an even more acidic medium, you speed up the growth of the roots. So this was pretty cool, because we really could show that alkalinization of this apoplast was sufficient to inhibit root growth inhibition, and this is what we needed. We needed a causal effect. So this also is cool, because what I said, pH and apoplastic pH more specifically makes sense, because I think it's from 1940s, there was already this acid growth theory going on that involves pH changes within the apoplast, because you will need a lower pH that activate enzymes to allow your cell wall to be more flexible. So what is actually happening is that what is said in this theory is that auxin stimulates proton pumps that pump out protons into the cell wall, where enzymes get activated. The cell wall is more flexible, which then would allow water uptake and other ion uptake that will make the cell swells, and swelling means growing. So proton pumps play an essential role in this model, and there is a pH effect as well in this model. So what we did was, well, actually we do see this alkalinization, but do we really see that when we check protons as well? So therefore, we actually took advantage of a reporter line that is, it's a genetically encoded reporter line. So it's fused to, what is it again? It's a membrane anchored protein element. So where you fuse a ratiometric, again, dye to. So it's a fluorometric dye that you can then use. So you can image this. So what is happening is, this is what I just showed you, the slide before, where you have the correlation between apoplastic pH and growth. But inside of the cell, where we have this genetically encoded marker, then inside of the cell, we don't see that at all. We actually see the growth is inhibited, but also the pH goes acidic, more acidic. So yes, we have actually something that makes sense, because we have more alkaline apoplastic pH and more acidic and intercellular pH. And that makes sense. So that would also mean that there is a change in the net proton fluxes within those cells. And we have a collaborator that manages to measure proton fluxes with non-invasive microelectrodes, and actually he could confirm that there's a net increase of the influx of protons in the cells. Now, you might start questioning this, no? Because I say proton influx, but this is all based on growth, where you have proton efflux, where that happens. Now, you have to remember, what we are looking at is the root. In the root, what is happening is that auxin inhibits growth. This is completely in contrast with what is happening and known and described much more in the shoot, where auxin is actually enhancing growth. So in the shoot and in all these coleoptile experiments, where auxin accumulates, that's where you have more cell elongation. So that's where you have more growth. In the root, we observe completely the opposite. So now we had these fast responses that we could correlate with pH and growth, but things still look too fast. And what is actually happening in the animal field is that for fast responses, very often phosphorylation or phosphorylation responses are responsible for the fast response. So what we could do was, and that was actually a lot of my input, is why didn't we do an auxin treatment in the root specifically and then go for a very fast phosphoproteomic research, which would mean that we isolate root tips, we filter them. So this is just filter-assisted peptides, doesn't really matter what it means. So we get peptides from these roots, we enrich the peptides, and then we throw them over titanium beads that will specifically enrich phosphopeptides. If we then analyze these phosphopeptides through a bit of a intensive software packages, then you actually would have specifically phosphorylated items within very short time frame of auxin treatment. And I say very short, it's actually 2 minutes, because physically I'm not able to cut root tips faster than 2 minutes to have enough material to actually do a mass spec proteomics approach. So it's 2 minutes. I probably, if I would be faster and the technology would be more sensitive, could do shorter, but it's 2 minutes what I have now. So what we have then actually is we have a very significant differentially set of proteins. Most of the ones in auxin treatments are actually hyperphosphorylated. So it would be cool to dig into this and find out what is happening there. But what we thought of was, as the auxin field is so well known, we should take advantage of what is known. And for that, I have these two parts here. We actually took advantage of the fact that there is a known anti-auxin, which is called POIA, so it's spelled like this. And if this POIA is within the receptors, so the nuclear TER receptors, then the co-receptor cannot bind to it. So in this case, you would have something binding to the receptor, but not stimulate the downstream signaling. So it's an anti-auxin. And then there's a second invention, and that's an orthogonal or engineered receptor pair, in which you actually have an engineered concave TER receptor. So it's this receptor, the normal one, that is just modified a little bit in a few amino acids. In order for it not to fit normal auxin anymore, it fits only this companion, the convex IA auxin. And this convex IA does not fit at all in the normal TER receptor. So by doing this pair, you actually specifically can induce what is happening downstream of TER1. I'm saying TER1 specifically because we don't know whether the other receptors might be binding it a little bit or not. So that means I did also the phosphoproteomics of a convex IAA in the concave TER receptor, and I did it also in the POIA. And also already here you can see that there's different responses. There's much more hypophosphorylation in these two treatments than there is in the auxin by itself. And if you then look at this, you really see that there's a lot of proteins that are uniquely affected by auxin and not by the other components, which could indicate that we are here really identifying items or elements that are independent of the known signaling pathway. So we have potentially a very interesting new set of data here. And then what we also did was we overlapped our set of phosphorylated proteins with a published phosphoproteome data set that is actually at 2 hours of treatment. And there you see that we are also hitting a very unique fast response. So this approach gave us potentially very interesting things. And then to go into the real interesting results is we really had differentially phosphorylated targets that would make sense with the fast growth response, because we had AHA proteins or proton ATPases as a target of this hyperphosphorylated phosphoproteomics approach. And what are these proton pumps? They pump protons, as I said, outside to the cells. In Arabidopsis, there's 12 of them, but AHA1 and 2 are most abundant and highly expressed in different tissues. They are rather essential for plant development. They really make sure that you build this proton motive force, which establishes or maintains the membrane potential and actually then affects all uptake and exchange within a plant. And they work like this. So in an inhibited state, they don't do anything, but when they are phosphorylated, there's actually interaction possible with 14-3 proteins, and there's ATP hydrolysis happening, and then the proton can be exchanged. So they are interested and related to pH. It was not something new what we found. I mean, within the stems and hypocotyls, what I said before, it is known that auxin has an effect on the activation of proton pumps. But you have to remember, I mean, this is within a stem. I said already there, auxin does the opposite effect. And it's also, no one there could cut it down into our time frame. So everything was later, much later than what we did. So what would be the mechanism of these in the hypocotyl where they are known to work? Again, I come back to this acid growth theory, so I don't really have to tell you what is happening here anymore. Protons go out when the proton pumps are activated, phosphorylated, and actually the pathway is known in the hypocotyl. It is through the sour genes, which are the first ones coming up when you have the TIR pathway happening. So these are transcriptionally coming up 2 to 3 minutes after the nuclear pathway is activated, and the sours inhibit a phosphatase. The phosphatase is then not removing the phosphate groups anymore. The proton remains active and it's pumping out. However, if you see that in a schematic view, there is a big part missing, because if you have a phosphatase, there should also be a kinase that is actually actively phosphorylating the protein of interest as well. And that, even in the hypocotyl where much more research was done, no one knew which kinase that could be. So as I said, auxin induces hypocotyl growth, acidification, and in the root does the opposite, and there it does alkalinization. So what we assumed was maybe the mechanism is maintained and it just works oppositely in the root. So as I said, we had this phosphorylation of AHAs from the mass specs data set, which you see clearly here. We had it nicely significantly phosphorylated, and we have it consistently in all our sets that we do. And we actually also see a slightly increased hydraulic activity, which means ATPases are really working. So they're really pumping. It's not just that it's active, that is phosphorylated, it's also activated. So that was very nice. We have this activation of the proton pumps, and we could also, in a very ugly blot, but we can nicely confirm that we actually have more phosphorylation of the activations, the known activation site of these proton pumps. So what you see here is no effect on the general protein level, but a clear increase over time of the phosphorylated site. So that is nice. It's for AHA2 that we have this phosphospecific antibody. So this is an easy Western blot confirmation. But as I said, we have this alkalinization happening, so the pH increases, but at the same time, we have a proton pump being activated as well, which actually would mean that you have acidification going on. And you can actually visualize that with this bromocresol blue. It's very ugly, but when it turns yellow, there's actually protons being pumped out of the root. So it's in your medium. So your medium changes pH, which means there's acidification going on too. And that means that probably we are faced with two antagonistic pathways that are both present at the same time, and they will balance each other out to allow for flexible responses, whatever is necessary at which time. And that makes sense in case of root growth. So if we want to gain more insight in this antagonistic function, we could actually chemically activate the proton pumps by using a fungal toxin. It's called physicoxin. That stabilizes the interaction with the 1433 proteins, and it would lead immediately to a little bit of a drop of the pH, which means we activate the proton pumps. We see a drop in the pH, acidification, that makes sense, and a promotion of the root growth. So simultaneously, we see a promotion of the root growth. So it looks like you really do have two separate things. Like when you activate the proton pumps separately from the auxin, you do see acidification. And then we have the other thing going on is, it's a bit tricky because in the proton pumps, you have 12 of them, as I said, so there's a lot of redundancy within the family. Mutants are not so easy. They are lethal very often. So the moment you combine 1 and 2, there's nothing happening anymore. You don't have fertile seeds anymore. So what we did was we have an siRNA targeting 4 of the AHAs, but specifically expressed from a root-specific promoter. So we express it under the promoter of PIN2, which we know that that's in the root very nicely expressed, and in the external cells of the root, which also makes sense for the responses of the root growth. And then from that promoter, we actually can reduce the AHA expression. So what we see in those mutants is we see actually that mutant is hypersensitive to auxin when you add it. So there's more alkalinization in this mutant, so higher values here than there is in the wild type. And there's less growth in it. So it's hypersensitive if you add it. And it's also hypersensitive if you would grow it on a concentration range of auxin. You always see more response to auxin in low concentrations than it would in wild type, which is the black graph, or in an it's a constitutively overactive AHA1 mutant. It's called open stomata 3. This one is always open proton pumps or always active proton pumps. There you have a reduced sensitivity. So again, this kind of makes sense that we have auxin-mediated activation, and that is antagonizing the auxin-induced alkalinization, because this would mean that we have alkalinization going on. And then this is back to what is actually responsible for this. There's an ongoing discussion within the field that it's probably not TIR and AFB signaling, and it's probably or maybe this ABP1 TMK1 pathway. Now, there could be indeed a role for TMK1, as I said before, and TMK1 belongs to a family of 4, so there's 4 of them. And what I had been doing was I grew, I think, 1,000 plates of roots of a TMK1 GFP line, and I looked whether I could find things that I can co-purify with this. So you actually can do a co-immunoprecipitation assay followed by mass spec, and you just do the proteomics of everything that you find that is co-purified with your protein of interest. Nicely enough, I had plasma membrane proteins, so I was really having something that had to do with transmembrane kinase, so a plasma membrane protein. And my 4th hit here were AHAs, which made me really think, like, if I have an interaction between TMK and AHA, then maybe I have in my hands the unknown kinase, because we have a transmembrane kinase that is interacting with it. I just did a few implanta experiments to really show that I have them interacting together. One of them is this bimolecular fluorescence assay, so BIFC. What you do there is you have either one construct with a split YFP, the other construct with the other side of the YFP. If they come together, you actually have reconstituted YFP. What I have to say is it's hyper ugly, but that is because when we overexpress TMK in tobacco plants, the plants shrink, the cells shrink, the leaves wilt. Plants really don't like this protein being overexpressed. What I then did to overcome this transient and weird system in tobacco, I had stable lines of TMK and AHA. And if I reciprocally pulled down my target, so either TMK here or AHA here, then I could actually detect the opposite partner. So I could detect AHAs in my TMK sample, and I could detect TMK in my AHA sample. So it's a confirmation that they were interacting in planta as well. And then I also always blot with this specific phosphorylation antibody, and I see that auxin still has an effect in both of the cases. So then I really wanted to show that we really do need TMK1 for this phosphorylation of AHAs, and I did a phosphoproteomics assay again, but now in the TMK1 mutant background, and I saw that every single phosphosite that we have in AHA proteins is downregulated. So normally you would have a phosphorylation level of 1 to be normal. These are all lower than 1, so every phosphosite is downregulated that I find. So there's hypophosphorylation of AHAs in the TMK mutant background. And this is my messiest slide, I'm really sorry for that, but what we do also do is we check whether we still have this induced phosphorylation in the mutant background. So in the TMK single mutant, we don't really see it anymore so significantly. So there's a really increase in phosphorylation here, and there's far less, we think, in this one. It goes better when you use double mutants. So the moment you go in a TMK1-3 mutant, you really lose the phosphorylation induction. And the TMK1-4 mutant, which is a very strong dwarfed mutant, has completely no phosphorylation anymore of the AHAs. So it really does show that you do need TMKs to be phosphorylating these AHAs. And then if you would do it in a activity assay, the hydrolysis assay, because AHAs do need ATP, then you actually see that only in Colombia you have this induction of hydrolysis, this increased activity. You miss that in all the TMK mutants. So interesting, we had really shown that TMK was the kinase responsible. And then we have actually a whole theory that there's these two antagonistic pathways. We have a lot of proof that we might need for half of the pathway the third still, even though there is no known transcriptional response that goes as fast as what we see. So we think that this side is, so that's under investigation now. People are really interested in this.
Inge, can you repeat the last sentence there? It just froze for a second.
Okay. Is it better now? Okay, so we have a lot of indications that we do need a third pathway for our responses, but probably not transcriptional, because it's too fast. So you cannot have transcription going on faster than 3 minutes. So this is under investigation. The group is very interested in this. And then we have this separate pathway that probably involves an auxin interaction with TMK ABP1. Even though that's also a paper that will come soon, because I investigated whether TMK could be the receptor for auxin by itself without need of ABP1, but it doesn't seem to be the case, sadly enough. But then actually you have this pathway that would inhibit the apoplastic pH, and then by this balance between the two, you actually can regulate growth. So this is the first paper we have. I think it's interesting, but we will see where it gets. Then the second one we have stems from the same phosphoproteomic data set. What we also found in there was actually a very fast phosphorylation of myosin XI. And that is a motor protein. It has actin binding capacity, and it's presumably involved in endomembrane vesicular transport. So why is this interesting? And that's because of this. I mean, I go back to this slide very fast, but it is because what I said, PIN transporters and the localization of them is essential for the whole transport of auxin within a plant. And the localization is determined very often by the recycling of them. So they go from inside of the cell, there's some signaling that tells them, yes, you have to go back to your basal localization, or there's some signal that says, wait, we need more auxin to the lateral neighbor, so you go to your lateral side. So that is the recycling really determines what is happening, and that's why myosin proteins are potentially interesting as well. So I found them phosphorylated in response to IE8 treatment, as you see here. So nicely phosphorylated, less in my mock, more in IE8. And I didn't see so much effect of my treatments, my other treatments. Remember, these were the showing whether it's stir dependent or not treatments. The effect is there a little bit, but it's far less significant, which made me think maybe this is very interesting, because I finally have something that is fully stir independent, which would mean that you have a completely novel signaling. We can confirm the phosphorylation of this myosin K protein in something that is called PhosTAC Western blots. In those Western blots, you have actually a molecule that is interacted with phosphorylated proteins. In a normal blot, a protein that is phosphorylated, you don't see. It would run in one line, but in a PhosTAC blot, you actually separate the phosphorylated moieties from the non-phosphorylated moieties. So you actually have a multiple bands. The only problem is the gels very often run smiley and ugly, but this is considered phosphorylation. So we do see, if you would quantify the ratio between non-phosphorylated and phosphorylated, the phosphorylation as well. And then within the myosin XI family, there's actually one member that is also very highly expressed within the root. It's the 11F member, and that one has actually very similar defects if you would look at what it does to the plant growth. So from now on, I will not just talk about myosin K, but I will also talk about myosin F. We have double mutants from now on. So how do we then determine whether there is defects in PIN trafficking or localization? There's a few experiments you can do, and I know also this has been controversial since our most recent publication, because you have a toxin, it's called brafaldin A. It's actually an inhibitor of ARF-GAFs, which are very important in vesicular trafficking. And when you use this one, you will actually see that PIN1 starts accumulating in bodies. So why did I in the beginning stress that we are able to do immunolocalization? That's because of this. So here what we do as well is we visualize PIN in plants without having a fluorescent PIN targeted there. We are really visualizing it through the endogenous antibody. So what's happening is that when you treat a plant with BFA, you actually start seeing that there's trafficking going on. However, the trafficking is blocked at some point, and there's accumulation in these blobs, and it's called BFA compartments or BFA bodies. So this is just a drug that can show you that pins are indeed being trafficked. So what you then do is, if you treat the plants with BFA and auxin, you actually see something very cool. And that is that there's suddenly far less of these BFA bodies, which means that auxin interferes with its own recycling. And that's what I said in this first slide. That is a known thing. It's visualized by an artificial drug, but it's kind of cool. Another way to visualize this is by using FM464. It's a dye that internalizes and binds to membrane structures. So you would see it in endosomes as well. So that's why in the wild type treated with this dye, you see nicely endosome compartments. So you see the dye there. But if you inhibit trafficking by having auxin there, then you have less of these endosome compartments. What we then did is you already don't see any of these BFA bodies in the myosin double mutant, and you see far less of the FM internalization in the myosin double mutant too. So it really looks like that auxin is interfering with its own transport, and that is going through the myosin protein. So the myosin mutant that doesn't have them is really required for the trafficking. We then did the same thing, but what we did now is, instead of just using myosin mutant, we made a myosin phosphodeficient mutant, which means we knew exactly which site was auxin responsible from the phosphoproteomics, and we mutated that site. So if you do that, you can actually mutate a serine amino acid to an alanine amino acid. An alanine will not get phosphorylated. It doesn't have any spot to get a phosphor group attached to it. And what we did see in that both in F and K mutant is that there's suddenly no BFA effect and no NEA effect anymore. So suddenly everything was gone, also for the FM uptake. So suddenly in these phosphodeficient mutants, there was no effect anymore. So it really does seem that this auxin-triggered phosphorylation is crucial for the function of the myosin as well. And it's crucial for PIN trafficking. Now, as I said, we look at two things that are a bit artificial. So we have a couple of better ways to look at PIN trafficking and trafficking in general with a more physiologically relevant output. For example, one of them is as I said, the basal to lateral relocalization of pins. That happens in a root when you treat it with this artificial auxin NA. When you have too much of that auxin, the root decides that it should get rid of this auxin and it should do something to the PIN proteins. So again, these are these immunolocalization images. In a mock-treated seedling, you do see the PIN 1 mainly on the basal side on the bottom of the cells, but in the NAA treated, it goes to the inner lateral side. So it pushes the auxin into the main vascular stream to get rid of it. That is not happening as much in the myosin mutant, and is also not happening anymore when we have the phosphodeficient mutant. So again, we show that you really do need this myosin phosphorylation to have normal PIN responses. And as I was speaking about more physiologically relevant responses, the shoot grows up. We all know that a tree faces with his leaves to the sky and the root goes into the soil. So a hypocotyl, which is on the seedling level, the shoot, does also do that. So when you switch a plant, so you actually are teasing a plant and you give it a turn of 90 degrees and suddenly make the hypocotyl face the new gravity response, the hypocotyl will actually start bending upwards. This also depends on PIN proteins because it are the pins that result in this asymmetric auxin distribution that allows this side to elongate and the inner side not to elongate. But at some point, when actually a 90-degree bending is reached, this hypocotyl should also stop bending. And that also, again, is because the pins reestablish polarity again. So what is actually happening in the myosin mutants is that they are not stopping when they are supposed to stop. So they are delayed, which means they are hyperbending. So if you would measure their angle, it's always much more than it would normally be in the seedlings. And you can also see that here they bend faster and they go to a bigger angle than they should have. So there really is a pin 3 re-establishment problem. The pin 3 stays asymmetric for too long. And that actually, as we show here, is also happening, or is also depending on the phosphorylation of myosin 11, F, and K. And then lastly, to be completely physiologically relevant, you have a process that also involves pin proteins, and that is when you cut a stem, you actually cut it in a way that you disrupt the normal auxin flow, then there's actually a way for the plant to recover from that. And it will actually make a channel to overcome this defect. And this channel is, of this canalization hypothesis, is all based on the most predominant pin there in the inflorescence stem, which is pin 1. So it's pin 1 that starts guiding this. I don't visualize pin 1 here, I visualize secondary balls. So this is a toluene blue stain that visualize the cell walls differently. So this is a real new channel because I had cut this original transport mechanism of the stem. In wild type, that happens within 7 days. But in my myosin mutant, within 7 days, I never see a fully formed channel that connects back to the tissue. So there is, again, something wrong with where the pins need to be and how fast they need where they are supposed to be. So there's never fully connection. Sometimes I have loose individual cells. I never see anything that is connecting like in wild type. I have the same thing happening in my phosphodeficient mutants, and it's even stronger. These ones, they don't even start differentiating cells here, so there's not even a channel being initiated when you have it in the phosphodeficient mutant. So I also always have this wild type supplemented lines as controls to have the same transgenic control in there. But then even more importantly, and that's why auxin is known for it, auxin has this self-organizing aspect of plant development, which means that when auxin accumulates somewhere, from there it will actually start initiating vasculature. That's what you see in the leaves, that's what you see in the cotyledons, that's what you see in this setup as well. So we again do the same thing. We cut the stem, we disrupt the vasculature, so no auxin is coming from the top anymore. But what we do is we apply in a drop of Vaseline, so very greasy thing, we apply auxin, and you actually do see that there is differentiation going on from this auxin drop to connect this to the vasculature. So it's kind of like the plant thinks that this is a new bud being formed and it has to connect it to the vasculature. We cheat because we cut it first, so but that's what is happening. This is also happening in when we supplement the plants with the wild type versions of this myosin mutant, and it's not happening anymore when we have the phosphodeficient mutants. So yes, auxin canalization is also impaired, and based on what I just showed you all, we really do think that this auxin-induced myosin phosphorylation is required for all developmental processes that require this coordinated pin-dependent auxin transport. This is very, very new, as people know that auxin is affecting its own transport, but the mechanism by which was not known yet. So we now would be among the first that actually have a direct regulation between an actin-binding motor transport like myosin and vesicular trafficking with an effect on pin transport. So to conclude, I can only thank everyone I've been working with. The list can never be too long. I have a very nice group. Jiří is of course my boss, and we collaborate with quite some people for phosphoproteomics approaches, like Bert Rebel in the VIB in Ghent, and then Dolf and Mark in Wageningen. And I would like to thank you for your interest, and maybe by questions you get this aha moment.
That was fantastic. Thank you, Inge. An excellent last slide. So if people have questions, you can either put them in the chat and I'll read them out, or you can unmute yourself and Inge can answer them. And maybe I'll start. I have a couple of questions, but the last few slides looked a lot like grafting, except for the canalization and in the upper left, the pin-mediated auxin stream that gets reset up goes inward in the stem rather than outward. So when we graft, we see vascularization around the periphery of the stem. Do you have any thoughts about that? Is that simply because those cells are still like symplastically connected, and if you were to sever it completely, the canalization would happen externally, or what are your thoughts?
Exactly. So what I do think indeed is that because we don't sever it completely, we don't cut it completely, that we don't force the plant to be smart. So we don't force him to go outside. So we have this inner connection possibility.
Neat. Yeah, we'll have to try that. We've never tried actually like a half graft before. It's a really cool idea. And then I assume this is an open area of research, but how do you think myosin XI phosphorylation is actually telling myosin how to polarize, and how that sets up the polarity of the pins?
So what we are currently trying and still failing at is to visualize Auxin-induced polymerizations, changes, or directional changes of myosin fibers within the plant, because that would indeed give us an answer. Now, if we would see auxin-induced changes, then that would tell us, okay, the fibers go somewhere differently or polymerize faster or slower, which would indeed correlate with a slower vesicle trafficking or different location targeted trafficking. That we sadly enough don't know yet, especially not like how that would affect the docking then eventually of these pin proteins in a different or the same where they originally came from membrane structure. That we don't know yet.
Soon, one day.
Yes.
I love how it connects to the gravitropic response and the opposite responses in roots and shoots. Very cool question. Well, does anyone else have questions?
Yeah, Margaret, I have a question. Yeah, great talk. I was trying to keep a few things in my mind during the talk, and one thing I was thinking about is kind of the physiological responses that you're looking at between pH and root growth rate. And if I remember correctly, it seemed like the application of the fusicosin prevented the change in pH in the root apoplast, but you also saw the reduction in the root growth rate. So, I mean, is that suggesting that the pH effects are just happen to occur in parallel, and that the auxin effects are actually independent of pH? Or maybe I missed something. Oh, okay.
Yeah, so what we do see is that by activating the proton pumps, you actually have a slight acidification. So the pH still drops a little bit. So that's this one here. No, it drops a little.
Okay.
It doesn't, it's not to the same level as normally the increase would be. So the increase would be double as high, but it drops a little bit by this activation. And the root growth does the opposite. So to enhance root growth.
Yeah, no, thank you. I was just trying to keep that in mind. I think I had the graphs flipped in my head. So,
yeah,
thank you.
I went fast over them as well. I'm sorry.
No, that's fine. And then one other question I had was just thinking about the confocal setup, and I'm just curious if you're using like MS media for that, and if you guys have done any sort of like chemistry of it, because I know sometimes the iron chelate and all that in the MS can.
Yeah, so we do use MS media for this, but depending on what application we are doing this for, we do the chemistry of the MS medium. So if we are looking at anything calcium related, we do not put calcium ions in the MS medium, or we try to avoid to have too many of them. If we do anything that like, if you have chelation going on, or if you change something, phosphates very often start to crash out of your MS, which means the pH drops immediately as well. So we try to keep it in mind, and we try to buffer everything we do as well. So every change we do, we add it to the medium, but we keep the medium buffered at our 5.8 pH. And we try just to keep an eye on it so that we don't overdo it. But yeah, it's a half MS based medium.
Great, thank you.
If I can, yeah, go ahead, Mrs. Harrison.
Hi, I'm Maria. Thanks for a great talk. It was really interesting. I had a question about the effects on cell expansion. So you've got this rapid growth and the wall loosening hypothesis, but there should be also an expansion of the vacuole in the cells in the expansion zone. And is that also controlled by auxin? So Turgor alterations.
Yeah, I can only talk about the root because that's where I checked it. There we don't see it. So we don't see that the vacuolar is actually constricted then because it's inhibition, so it's the opposite effect. So we don't see it in the root cells.
Okay. Thank you.
There's a question in the chat. Very cool research. What could be the proposed mechanisms that would lead to proton influx in the root in such a short time frame? By proton channels?
Proton channels, but they're not known. You have symport going on. So, for example, the auxin importers symport 2 protons with every auxin molecule it takes in. So that could be a reason why we see it so fast, because auxin comes in and the protons come in together with it. But this mechanistic question is what is going to keep us busy for a bit longer, because we don't know. And there's potentially link with calcium influx and outflux. So potassium is also probably related. So this is much more complicated. Now, the moment you start switching ion responses, you trigger Turgor responses, and then you have everything maybe triggering the other thing.
Yeah, it's complicated, but I think you took a deep dive in, figured out some of the mechanisms. Oh, that's great. So I think we're over time now. Sorry about that. So let's thank Inge for a wonderful talk. Thanks for wrapping up our seminar series for the semester. And I hope to see you in person sometime soon.
Yep. Yeah. I'm available for students later if needed.
Yeah, great.
Bye.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.